Cells are surrounded by different types of solutions. A solution is a homogeneous mixture of one or more solutes dissolved in a solvent. Solute is a substance that is being dissolved, while the solvent is a dissolving medium. If you mix sugar in water, the sugar is a solute and the water is a solvent. A cell is basically a solution inside a partially permeable cell membrane. Substances move in and out of the cell through the cell membrane. The substances that move in or out of the cell can be water, salt, and organic molecules. For example, water moves from a high concentration of water particles or a dilute solution to a less concentrated solution of water particles or a concentrated solution across the membrane of the cell. Solutions surrounding a cell can be isotonic, hypotonic, or hypertonic solutions. An isotonic solution is when two solutions, separated by semi-permeable membrane, have equal concentrations of solutes and water. Hypotonic solution refers to a solution that has a lower solute concentration compared to another solution. Hypertonic solution refers to a solution that contains a higher concentration of solutes compared to another solution. Cells react differently when they are placed in isotonic, hypotonic, and hypertonic solutions. If a cell, plant or an animal cell, is placed in an isotonic solution, there will be no net flow of water into or out of the cell, and the cell's volume will remain stable. This is because the solid concentration of the solution is the same as the solid concentration inside the cell. Isotonic solution has an advantage in different organisms. For example, in humans, when the plasma surrounding blood cells is an isotonic solution, compared to the solution inside the blood cells, the cells function normally. The isotonic solution allows the cells to move water and nutrients in and out of the cell. This is important for blood cells for delivering of oxygen and other essential nutrients to other parts of the body. In some cases, the solution outside the cell can be hypotonic to the cell. If a cell is placed in a hypotonic solution, there will be a net flow of water into the cell, and the cell will gain volume. This is because the concentration of the solution outside the cell membrane is lower than the concentration of the solution inside the cell. As a result, water enters to the cell through the cell membrane, and the size of the cell increases. Animal and plant cells show different reactions when they are placed in hypotonic solution. If red blood cell is placed in a hypotonic solution, water enters to the cell and the size of the cell increases. Finally, the cell ruptures. If raisin, dried grape, is placed in hypotonic solution, water enters from the solution to the raisin cell. The size of the cell increases and finally it becomes turgid. Unlike an animal cell, the plant cell that is placed in hypotonic solution does not rupture. This is because plant cells have a rigid cell wall around the plasma membrane. The cell wall prevents the cell become rupturing. Upon swelling with water, the cell becomes turgid. Different organisms living in the ocean get salt in their body from the food they eat or from the water they drink. These organisms have different mechanisms to remove excess salt from their body. For example, whales get salt from the food they eat. in order to remove the excess salt from their body. Whales have salt glands. The salt glands extract the excess salt from the bloodstream and excrete it through the nostrils. This mechanism is important to remove excess salt from their body and maintain hypotonic environments, and enables the organism to survive in concentrated habitats. In some cases, the concentration of the solution outside the cell can be higher than the concentration of the solution inside the cell. Such solution is a hypertonic solution. If raisin, dried grape, is placed in a hypertonic solution, since there is a low amount of water in the solution than the cell, there will be a net flow of water out of the cells of the raisin, and the cell will lose volume and becomes shrink. This process is known as plasmolysis. Plasmolysis is the shrinking of the protoplasm of a living cell due to loss of water. If an animal cell, for example, red blood cell, is placed in hypertonic solution, water moves from the cell to the solution. The cell loses water and undergoes shrinkage. This process is known as crenation. Water moves out of the cell because the concentration of the solution is higher than the cell. The human brain plays a role to regulate the amount of water in the blood. If the cell environment becomes a highly hypertonic solution, it is because there is not enough water in the blood to dilute the solutes. Therefore, to regulate the amount of water, the part of the brain called hypothalamus releases hormones while increasing the permeability of membranes in the kidney. As a result, the kidney reabsorbs the water that would have been excreted and adds it back to the bloodstream. The blood becomes more isotonic compared to the cells, and normal process can continue. Let's look at the following experiment on hypertonic, isotonic, and hypotonic solutions. To conduct the experiment, the following materials are required: 6 beakers, potato, cork borer, ruler, water, salt, timer, digital weight balance, knife, pen and paper. To start the experiment, follow the following procedures. Using cork borer, prepare 18 potato strips in equal lengths, 6 cm each. Put 3 potato strips in each beaker. Weigh salt. Prepare 6 solutions of salt and water having different concentration. That means solution 1, 0 gram per 100 milliliter, solution 2, 1 gram per 100 milliliter, solution 3, 2 gram per 100 milliliter, solution 4, 3 gram per 100 milliliter, solution 5, 4 gram per 100 milliliter, and solution 6, 5 gram per 100 milliliter. Pour the solution in each beaker. Leave the solution for 20 minutes. After 20 minutes, remove the potato strips and measure using a ruler. And finally, record the data. The result of the experiment indicates that the potato strips immersed in higher concentration solution becomes soft and decrease in length. The potato strips immersed in lower concentration become turgid. From the table, as you can see, the initial length of all the strips was 6 cm. The final length and the percentage of change in length of the potato strips varies. When we put these values on the graph, we can find a downward curve. The y-axis of the graph shows the average change in length in percentage, and the x-axis shows the salt concentration. The data from this graph shows that at low salt concentration, the average change in length of the potato strip is increased. For example, when the salt concentration is 1 gram per 100 milliliter, the percentage of the average change in length of the potato strip is 1. This is because water has entered to the potato cell by osmosis. This results in an increment in the length of the potato strip. This indicates that there is higher water potential inside the solution than the potato cells. That means the solution is hypotonic solution. At high salt concentration, the average change in length of the potato strip is decreased. For example, the percentage of the average change in length of the potato strip that is placed in the solution with salt concentration of 4 gram per 100 milliliter is approximately below 0, 4.4. This is because there is higher water potential in the potato cells than the surrounding solution. As a result, water leaves the cell and enters to the solution. This leads to a decrease in length of the potato strip. That means the solution is hypertonic to the potato cells. At the point the graph crosses at 0, where the concentration of the solution is about 1.4 gram per 100 milliliter of salt, the solution is isotonic. In this case, the water potential inside the potato is equal to the water potential in the solution. At this point, there is no net movement of water. In general, if a plant cell is immersed in isotonic solution, there is no change in volume of the cell. If plant cell is placed in hypotonic solution, it gains water and becomes turgid. If a plant cell is placed in hypertonic solution, it loses water and decreases in size. In human body, the normal blood concentration is 99.9% of water and 0.1% of salt. The concentration inside the red blood cell is the same. A patient is given a solution of 80% of water and 20% of salt through his vein. Based on this information, attempt the following questions. What type of solution is the red blood cells in after receiving the solution? A. Hypotonic. B. Isotonic. C. Hypertonic. What will happen to the red blood cells after receiving the solution? A. Swell. B. Shrink. C. No change in size of red blood cell. In human body, the normal blood concentration is 99.9% of water and 0.1% of salt. The concentration inside the red blood cell is the same. A patient is given a solution of 80% of water and 20% of salt through his vein. Based on this information, attempt the following questions. What type of solution is the red blood cells in after receiving the solution? A. Hypotonic. B. Isotonic. C. Hypertonic. The solution contains large amount of solutes as compared to the concentration of red blood cell. Therefore, the solution that the patient is given is hypertonic to the red blood cell. The answer is hypertonic, or C. Red blood cell is an animal cell. When an animal cell is immersed in hypertonic solution, it loses water and becomes shrink. The answer is shrink, or B. Cell's environment can be hypotonic, hypertonic, or isotonic to the cell. If an animal cell is placed in hypotonic solution, it bursts. On the other hand, if a plant cell is immersed in hypotonic solution, it becomes turgid. If cell is immersed in hypertonic solution, the cell loses water. If cells are surrounded by an isotonic solution, there is no net movement of water into and out of the cell. In general, the different types of solutions have their own importance to the continuation of life on Earth.